Good morning, viewer. Welcome to our Glory Globe TV breaking news. Viewers, long-term genetic tracking of patients with chronic blood cancers is revealing hidden patterns in how these diseases develop and progress. Patients over time has revealed key genetic differences between blood cancers that remain stable and those that become more aggressive. So the findings also show how DNA data could sharpen diagnosis, improve how patients are monitored, and give doctors a clearer view of how treatments are working. Together, this points to a future where routine genetic testing plays a larger role in standard NHS cure. In a study published in Cancer Discovery, scientists from the Wellcome Sanger Institute and their collaborators examined both genetic data and clinical records from patients with chronic blood cancers. By tracing the lineage of blood cells, they explored how these diseases develop over decades. So the results are also being presented at the American Association of Cancer Research Conference in San Diego. Myeloproliferative neoplasm, in the short form MPNS, are one of a form of chronic blood cancer. These are rare disorders begin in the bone marrow, where blood cells are produced in excessive and uncontrolled numbers. About 40,000 people in the UK are living with MPNs, and roughly 4,000 new cases are diagnosed each year. These conditions, viewers, tend to progress slowly and are driven by mutations or changes in DNA that can rise early in life and build up over time. Most MPN cases are linked to mutations in the ZAC2, CUR, or MPL genes. However, around 10% of patients do not carry any of these genetic changes. But in these cases also, diagnosis often relies on how bone marrow cells appears under a microscope. So as a result, some patients may receive treatment such as chemotherapy without clear genetic evidence of cancer. The course of these cancers varies widely. Some patients remain stable for years and need only minimal treatment, while others develop more serious conditions such as leukemia or scarring in the bone marrow. Doctors will struggle to predict which path a patient disease will take. To address this, the research team investigated whether genetic analysis could help forecast disease progressions and clarify whether patients without common mutations truly have cancer. They followed 30 patients with chronic blood cancers, mostly MPNs, combining whole genome sequencing with detailed medical records. The dataset included nearly 8,000 blood test results along with treatment histories and disease outcomes. In total, more than 450 samples were analyzed, with some patients monitored for up to 25 years during routine care. By linking long-term clinical follow-up at Cambridge University Hospital NHS Foundation Trust with genomic analysis at the Sanger Institute, the team tracked how blood cells populations change over time using DNA to build cellular family trees, and they identified the origin of cancer clones, groups of genetically identical cells that eventually drop disease progression. Amazing, sir. Bow down, bow down. Behind this exclusive report.